So Cliff, I think we're up to about 36 people. That's usually about what we get to. So feel free to start whenever you'd like.
Great, thank you. Okay, so thank you for coming. So today I'm going to talk about the functions of the Cistrome data browser and also the data that we have and the whole idea. So the format of the talk is I'll give a brief introduction, and then I'll talk about the Cistrome data browser. And then there are several applications which can help you find data like from the data browser, which are the toolkits, Cistrome Go, Lisa, and then I will conclude. So there's some basic questions which the Cistrome data browser can help biologists address. So the first question is, what are the publicly available high quality data sets that are available of some kind of TF or histomodification of interest? And so for this, there's, you can query the Cistrome data browser. Other questions include what transcription factors regulate a particular gene interest? What are the transcription factors that bind to a particular genomic interval of interest? And what are the transcription factor profiles which are similar to a list of genomic intervals that you might have? And these are functions of the Cistrome data browser toolkit. And then there are some other questions related to regulation of genes. So, for example, you might be interested in knowing what are the target genes of a Chip-seg peak set? What are the transcription factors? What is the transcription factor regulating directly? And what is the function of a transcription factor based on the Chip-seg and gene expression? And so for this, we have a tool called Cistrome Go. And then finally, if you just have a set of differentially expressed genes and you want to know what transcription factors regulate that set of genes, we have a tool called Lisa, which can help you answer that question. Okay, so with that, I will just give a brief overview that we all know that gene regulation in humans or mammals is very complex, involving like hundreds of different transcription factors. And there are hundreds of different cell types. And there are these epigenetic layers of control. And it's a lot of information and a very complex problem to understand. And for this reason, biologists all over the world have done these chromatin profiling experiments, including Chip-seg, DNA-seq, Attack-seq, and other chromatin profiling experiments, which can help to address these questions of how genes are regulated in higher organisms. And so these gene sets have really accumulated over the years, and people are still doing lots of experiments, and it's like they're kind of accumulating in repositories such as Geo. Problem with Geo is that it's kind of hard to know what's inside Geo, and it's hard to do an analysis based on the data in Geo because it requires so much effort to like map and process the data before you even know if it's high quality data. And so, so on the one hand, we have this, some might think, well, it's great, we have all of this data. We should be able to answer these questions if we can only find the right thing. And others might think, oh, well, a lot of it is, garbage. And how do we actually find this data? And even if there is data which we might think is relevant, it might turn out like a, in a garage sale, like a jigsaw puzzle with a couple of missing pieces. And so it's not very helpful. And so for that reason, we developed the Cistrome data browser. And what the Cistrome data browser does is it collects data from the major sources of these kind of chromatin profiles, including Geo, ENCODE, and Roadmap. And we have a pipeline to process the same data types by the same pipeline. So for example, Chip-seg will be, TF Chip-seg will be processed by one pipeline, and there'll be another for DNAs, but all the DNAs will be processed by the same pipeline. And so what the Cistrome data browser provides is data, as well as a means to analyze the data, to find the data, and also to do some visualization of the data. And our data includes both human and mouse data. And I will explain this functionality. So the uniform analysis pipeline was a pipeline we initially called Qilin, and now it's called CHIPS, which is the same as what the CFC is using to analyze their data. And so in this basic pipeline, there's the steps of aligning reads, calling peaks using max, doing some quality controls, calculating, for example, number of reads and peaks, PVC, looking for contamination, fast QC analysis. And then also there are some other kind of downstream analyses, which are simple downstream analyses of just looking at what are the distributions of peaks in different genomic features, such as promoters or exons and so on. And we also do motif analysis and find the targets of transcription factor binding sites. So the way you find data, the first way is to search for the data by keyword. And so this is from Data Browser. You have this toolbar where you can enter a keyword and it will find the factors that it matches. You also have the option, this is a sort of pro tip, if you know what the Cistrome ID is, you can just enter the Cistrome ID, and that's another way to find something. This can be quite handy sometimes. You can specify whether you want to find data only in human or mouse. You can specify the biological source, and then you can find the factors you are interested in. So here's just a note of caution that the way, because there are so many thousands, there are tens of thousands of different samples, we can't curate this manually. So we have a parsing script to curate it. And so we have to rely on whatever the people put in Geo to describe their samples by matching keywords and looking for serial numbers and so on. But there are often these sort of incomplete or ambiguous descriptions which can result in this annotation not being what you think it is. And in addition, you can also imagine complex experiments. For example, someone might knock down one transcription factor and do the Chip-seg of another. It can be hard for a parser to know which is the transcription factor being chipped and which is the one being knocked down. And there are lots of possibilities to make it look very complicated. And so for this reason, we provide links to Geo so that you can check exactly what the sample you're looking at is. And we also provide a form where users can give feedback to correct annotation if they do find an error. So one example of this is, for example, if you find, for example, estrogen receptor in your list, you can click on the bar, and then below this window, this inspector window will show up. You just might need to scroll down. So the inspector window will show you the title of the sample in Geo, and also give you a Geo ID, which you can click on. And then, so for example, the Geo ID might show you might be something like this. They did an experiment. They show the antibody is estrogen receptor, which is reasonable enough. But then they also include information about the extraction protocol, and then they mention 4 different antibodies, one of which happens to be CTCF. And so in this case, we're lucky that they put in estrogen receptor, but in many cases that you get this situation where this happens and there's no clear link to what the true target is. And so it's always a good idea if you really want to follow up to look at the annotation in Geo. And we provide the tool to do that. We also provide these quality control metrics. So one of the things when you look at the data, often or fairly often, the data isn't all high quality. And so we provide 6 different quality control metrics so that you can assess at least which ones are the better quality samples and which ones are not so great. So 3 of these metrics are related to the read. So the first is the sequence quality score, second mapping ratio, and the third the PCR bottleneck coefficient. These are related to the quality of the sequencing and sort of PCR amplification artifacts. And then the next three QC criteria are the number of peaks detected, the fraction of reads in peaks, and the fraction of peaks in DNA's hypersensitive regions. And so these are actually really pretty important. That's if you, for example, you only see 10 or 100 reads in your sample, that's an indication that the Chip-seq really isn't working very well. If the fraction of reads in peaks is low, then that's also not a good sign. And also if the DNA is the peaks are not falling in DNA's hypersensitive regions, previously found DNA's hypersensitive regions, that's also an indication of a poor quality sample. And so we just make it simple, pass, fail. And so you can look into the details on the website what pass, fail mean. But in general, like if you're seeing a lot of red, particularly in these peak QC metrics, that's a bad sign. I wouldn't worry too much if you see, for example, a poor sequence quality score, mapping ratio, so long as you see good QC metrics in these later criteria. Sorry, was there a question? Yeah, please, if anyone has any questions, please ask.
Hey Cliff,
yeah.
This is Ramesh.
Hi, yes.
Is there some way you could indicate on the Cistrome DB page which criteria we should pay more attention than others? Because that's not always intuitive.
Sure.
This is helpful to hear you walk us through it, but when we're actually working on it, it's not as clear.
Sure. Yeah, that's a good idea. We can discuss exactly what would be the best thing later at some other point maybe. But yeah, that's a great idea, because I think people don't realize that you might see that red on this side, but they're different, I think. Yeah. And so we because some samples you might have many, for example, estrogen receptor, you have several hundred samples. That you might have many of them. You might not want to scroll down to look at all of them. And so we provide these options where you can show only the samples passing either the peak quality control, which is these 3 on the right, or the samples passing all quality control. And so then if you have an abundance of things to choose from, you might as well just go for the high quality ones without bothering to look at the poor quality ones. Sometimes you don't have that option, and so you just have to try your luck with the bad quality samples. So we also show more detailed information on the QC report, so exactly what these different metrics are. And we show these fast conservation profiles. And so generally for a transcription factor, it's good to see a peak right in the center of this conservation profile, like right at this, like at the center of the peak, you should see the highest conservation. But sometimes, for example, for histone modifications, you see this kind of pattern where you see a trough of conservation and you see the peaks at the side. And that's because, of course, the histone is this is a nucleosome, and the true regulatory regions are flanking the nucleosome. They're not concentrated on the nucleosome. And so that's why you see this trough kind of pattern. And then just a final sort of word about the QC metrics, that these QC metrics are really focused on peak QC metrics. And so they're good for factors which have these narrow, sharp peaks, which include most transcription factors, DNA-seq and attack-seq, and also these histone modifications associated with active marks, for example, K4 monodi trimethylation, and K27 acetylation. But there are some Chip-seq samples, for example, these histone modifications associated with a sort of repressive chromatin, K9 methylation, K27 trimethylation, and so on, where these QC metrics are not really the best thing to evaluate the quality. And unfortunately, we don't really have a great QC metrics for these things. And in addition, we've also noticed chromatin-associated factors, for example, SP140. It seems to be not the same as a sharp peak. And so for those kind of samples, the QC metric might not be appropriate. And so then you would have to do some other evaluation to assess how that sample is. We do some DNA motif analysis. This motif analysis is based on the location of the motif relative to the peak summit. So what we're looking for is significant motifs which are kind of located close to the center of near the summit rather than uniformly distributed across the region. And we provide these reports in the web page where we group the TFs with similar motifs together. If you click on these links here, they show the actual motif. And it's always a good idea if you're looking at transcription factor Chip-seg and that if the motif is known, it's always good to see that the motif is maybe not number 1 on the list, but it should be on the top of the list, near the top of the list. If it's not near the top of the list, there may be a sort of, it might be the wrong antibody or something. Then we have this option of visualizing the data either on the WashU or UCSC Genome browsers. And so you can visualize multiple samples. You just check these boxes of the samples you want to see, and then click on the WashU browser. You have to be careful. You don't want to click on a mixture of human and mouse samples, then it won't work. You have to click on the same species.
So I find that the browser links often don't work.
Yeah, that's it.
I don't know if the local instance of the UCSC is not correctly configured, or often that click doesn't work.
Yeah, we need to check into what the problems could be.
Whether, I mean, we have a local UCSC instance, right, or not?
Not UCSC. I think we used to have a local WashU, but we not anymore.
I see.
Mm-hmm.
I think it's something we need to sort of check on.
And this is the kind of the thing you will see if it works. That you can see all these different tracks, and you can scroll and look at the data. So then, so that is the Cistrome data browser. And then there are several applications which interact with the data browser and use the data. And they help investigators answer certain biological questions. So the first tool I'm going to discuss is the Cistrome data browser toolkit. And so this addresses 3 questions. The first is which transcription factors regulate a gene of interest. Second is what transcription factors bind to a genomic interval of interest. And the third is what transcription factor Chip-seg profiles are similar to a list of genomic intervals. Sorry. So to address this question, what regulates your gene of interest, we use this thing we call a regulatory potential model. So this is what I think is the most kind of natural way to associate a TF and TF binding to a promoter. And that is based on an exponential decay weighted distance, which associates multiple peaks with a promoter. And so there's one parameter you have to fix, and that is the sort of rate of decay. And so we have the option in the toolkit to set this rate at either 1 kb, 10 kb, or actually 100 kb. And so what this will do is it will assign peaks to the promoter, weighting them by the weight function. So for example, in this example, this nearby enhancer will get a high weight, but these 3 will also be weighted and given lower weights, and they will be summed up, and that will be the regulatory potential for that gene. And so what we do in the toolkit is actually, we calculate these regulatory potential potentials for all high quality transcription factor data sets. But then there are genes which are in regions which are more highly bound than others. And so then we normalize the data by the median value over all of these different transcription factors. And that's how we rank the likely regulators. And so if you go to this, this link, dbtoolkits.cistrome.org, this is the first option. Very simple to use. You specify the species. You type in the gene symbol you're interested in. You can decide whether you want to look for transcription factors or chromatin accessibility, and then you set this decay distance and then submit. And then it will return a page like this, like a table, which is ranked by this relative regulatory potential. So if you ask what regulates the androgen receptor, it will return this list of factors. And again, you have the same functionality where you can now visualize this data on the WashU genome browser. And you can also inspect the data as you could with the Cistrome data browser. The same kind of interface. You can assess the quality of the data. You can look at the geo. You can do all of that kind of thing. The other views that you have of this data are these figures. So the one is a dynamic plot and the other is a static plot. So this just shows on one plot this information where the different dots are different samples. And you can download the figure as well. And so if you kind of change the regulatory decay distance, then you will get different answers. And I think it's a biologically meaningful. There will be some things binding at the promoter, which you could say do regulate the gene. And there are also things binding at enhancers, which also regulate the gene. And so it's useful to look at like both of these, like all of the regulatory distances to get a sense of what's going on. So the second function of the toolkit is answering the question, what regulators bound an interval of interest? And so instead of putting a gene, you put a interval, genomic coordinates, and it gives you also similar kind of feedback. For example, you can enter the coordinates of this enhancer region, which is described in this paper, and then it will show you AR, FOXA1, all of these factors are binding in that region. And you can go and visualize this as well. So what's also quite convenient, if you're interested in a SNP, you don't have to look up the genomic coordinates of the SNP. You can just put in the SNP ID, and you can also very quickly get to what's located on that SNP region. And the final function of the toolkit is what Cistrome data browser Cistromes are similar to yours. So in this case, what you have to do is you have to upload a file, which is in embed file format. So basically, I'm sure you're probably familiar, just the chromosome, start site, end site, tab delimited file. You can compare this either to the top 1,000 peaks in the Cistrome for each sample, or the top 10,000 peaks for each sample if they exist. And then you submit, and then it will give you feedback of what's similar to your set of genomic intervals. And so there's several kind of scenarios where this might be useful. For example, you might have a Chip-seg peak set, and you might be interested in other transcription factors with similar binding profiles. You might have done some attack-seq experiments, and you have some differential attack-seq regions. And then you might want to know what transcription factors are binding in these regions. And so you could just upload the file of the differential attack-seq regions, and then you could get some ideas of what the TFs might be which regulate that. So that's a pretty useful function. And then, for example, you could also have a set of K27 acetylation peaks.
Cliff?
Hello, yeah.
So just a quick comment to make that I completely agree with you that examining differential peaks with this approach is very useful, and we've built that into a recent pipeline we put to actually analyze differential peaks. So
yeah, that's great, and also single cell, similar kind of thing. You can, you know, it's a very helpful thing. And then the final thing is, you know, a K27 acetylation. They might not be differential, but you might not know exactly what kind of tissue this is. So you can try to say, you know, either the differential or the highest peaks. You can try and find the sample which is most similar to yours, and maybe it can help you to annotate what cell type you have. So this actually, just to mention that, like, this function can find either transcription factor Chip-seq, or it can also find DNAs and histone modification, and both can be helpful depending on the question. So this is just an example. If you put an input of a BATF, and you can search it either using the top 1,000 peaks or all peaks, it will find all of these other kind of immune-related TF Chip-seq samples. And works pretty well. It doesn't seem to be super sensitive on which option you choose.
So could I ask a question, Cliff?
Yes, sure.
So just to clarify, perhaps it's obvious, but this is based upon existing Chip-seq or TF Chip-seq data that's available in Cistrome. And so we're not going to find anything new this way. We're going to just compare it to existing databases. And so this does nothing to do with motifs. It's not looking at motif sequences.
Right. Yeah, so it is comparing with available data, and I guess it's sort of open question of how much would be new. I think, you know, it sort of remains to be seen. I think, you know, maybe once we start including single cell attack-seq, we might start seeing new things or something a bit different.
All right, good.
So, by the way, David, Cliff is leading a project with a student in the lab to impute transcription factor binding. This is using the epigenetic data, but use information more than just the motif to predict transcription factor binding. We are hoping to get, you know, most of the cell types, if the TF is expressed, having a prediction there. And then in the future, maybe we will also search for similar data sets of, say, if you have a single cell attack-seq, you have a cluster. And based on that, we could figure out what different Chip-seqs are binding in that cluster using the imputed data, but this will be a later feature.
Very good, thank you.
Okay, yeah, great, thanks. So the next tool I'm going to describe is Cistrome Go, and I think maybe a lot of people haven't heard of this. So Cistrome Go is actually, has a functionality which is a combination of what used to be called beta, which people know, which is a way of associating target genes with Chip-seq peak sets. And it's also has a functionality of what many people use, GREAT, which does the gene ontology analysis of Chip-seq peaks. And so, of course, these answer the question, what are the target genes of a user-provided Chip-seq peak set, and what are the potential function of a transcription factor? And the nice thing about this tool is it can also combine the analysis of Chip-seq and gene expression data together. So this is Cistrome Go. So in Cistrome Go, what you do is you input Chip-seq peaks, and the first thing that it does, it kind of makes a quick assessment of whether your peaks are mainly in the promoter or mainly outside promoter, or let's just say highly enriched in the promoter or not so highly enriched in the promoter. And this is a threshold, which is the default of 20%. You can change it to change your analysis. If you're doing a promoter type analysis, then it calculates regulatory potentials based on this 1 kb, this decay distance. So it's really only looking at the promoter. And for several transcription factors, we found that actually the regulation seems to be really concentrated at the promoter. And so it really makes sense only to look at the promoter for those factors. For other factors, like, for example, AR, ER, these kind of things, they are more enhancer related factors. And then we use this 10 KB by default, although the user can also change those defaults if they want to look broader than that. And then what we do is we normalize these regulatory potentials based on other Chip-seq data in the Cistrome data browser. And based on this normalized regulatory potentials, we do the go-term enrichment. At this point, you also have the option of, you can also put in input, which is differential gene expression. And so this would combine the target gene allocation together with the differential gene expression levels. And so after the integration, then you can do the gene ontology enrichment analysis. So it works pretty well. It's go.cistrome.org. It's really very simple to use. Just you choose the species, you're allowed to put in 38 or HG19 if you want. You put in the peak bed file in the bed format. The gene expression file can be either a DEC format or a simple format. It's just, you can get the answer. It's very easy to do that. And then you can specify whether you're interested in upregulated genes, downregulated genes, or if you just want to lump up and down together. And then there are also these advanced settings where you can specify these things of whether you want to change the regulatory distance. And so we have some demo data sets. So if you go to the website, you can look at the demos to see what kind of output you get. The solo mode is when you're doing the analysis based on the Chip-seq only. And then you also have this ensemble mode where you combine the Chip-seq with the gene expression. And so here you can look at the results from these different analyses to see what you can get from this. So this is one of the result pages. You get these regulatory potentials. And so this is actually, if you're used to using data, you can also do this. It's a little bit more flexible, and it also has this adjusted RP score, which is kind of using other Chip-seq, other TF Chip-seq data. So this can be more accurate than just the raw RP score. So I would actually recommend doing that. And then this gives you the ranking of the genes by RP, with coordinate, and then you can actually go visualize inside the WashU genome browser to see where your peaks are relative to that gene. And then you also have these gene ontology analyses. It gives you, similar to other gene ontology analysis tools, the enrichment, the p-values, and then you can also look at what exactly the genes are that it's calling inside that category and do some visualization as well. So actually, if you're using grade a lot, I think also you might want to try this tool. I would say it's, in general, working, they're kind of on a par, although like sometimes one works better than the other for reasons we don't fully understand, but sometimes this can work a lot better than grade. So it's definitely worth trying using this tool. Okay, so then the last...
Cliff, may I ask a small question? So we haven't used some of the more recent ones you talked about, but we've gotten a lot of value out of beta. And probably cited that paper multiple times and used. I wonder if there's any move in the group to make the output from beta a little more user-friendly, a little more intuitive, and also a little sexier. The way it currently puts out, we include it in papers and things, but I'm not sure that reviewers or readers fully grasp it.
Yeah, so I think we actually, Ramesh, I think, yeah, we should have a look at the Cistrome Go together, just to look at that output, and we can sort of evaluate that looks. Because I think beta, a lot of people had some troubles using, and so I think we'll probably use Cistrome Go instead of beta. But we can have a look to see if this output is an improvement or not. Yeah, so definitely, I think the beta, those beta plots were not completely intuitive, and but we could to reevaluate.
Thank you.
Yeah, so the last tool I am going to describe is Lisa. So Lisa answers this, or the question it can try and address is, what transcription factors regulate a set of differentially expressed genes? And so Lisa, actually, we've already kind of discussed what the peak, the regulatory potential is based on TF Chip-seg peaks. And so this is a way to quantify the association between transcription factor binding with transcription start site of genes, or just genes in general. We believe that the transcription start site is the important location to focus on. And it integrates the proximity of the transcription start sites to the binding sites, and also the number of peaks. When you have signal coming from histone modification data, for example, K20.
Quick question for you.
Question?
3 weeks time?
Okay. Sorry, is that a question for me, or? Okay, so if you have K27 acetylation Chip-seg, often the signal is not really confined to peaks, but can sometimes be spread around. And so then it's not really clear that you should associate the peaks of this with the transcription start site, or whether it's just the signal in general. And so for this reason, we calculate this what we call chromatin RP score, which is really, instead of just calling peaks and doing this calculation, we just associate the reads, or the signal, with the transcription factor binding site. In other sense, it's exactly the same thing. You're just taking a weighted, you're just weighting the reads according to the distance from the transcription start site. Okay, so actually this methodology was described in a much earlier paper called MARGE, which was published in Genome Research. And the idea was really that just to say, well, how do you quantify K27 acetylation signal? And what was popular at the time, and maybe still popular, as a super enhancer analysis. And so one of the claims of the super enhancer analysis was it could identify, important tissue-specific genes. And so actually we just did an analysis of, in terms of enrichment of the Rose super enhancer analysis relative to our relative regulatory potential analysis. And we find that actually our analysis it looks stronger than the super enhancer idea. So the lower triangle is like our p-value calculation, and the upper is the super enhancer calculation. And you can see, for example, in these sort of gene ontology categories, T cell activation, and these sort of all these blood-related things, you can see that the high scores you're getting from the relative the blood types. So it's exactly where you should see them. When you look at muscle or heart development, you see that it's like in the right type of tissues where you see the high scores. Where you see this sort of where Rose or the super enhancer is giving higher scores than what we see, it's kind of often in places where it's not really the right tissue. And so we believe from this point of view that this kind of analysis is better, a lot better than the Rose analysis. The other thing is, like, the super enhancer analysis was, supposedly the response of these BET inhibitors, for example, JQ1, that the super enhancers were supposedly the thing that could predict the genes which would respond to these drugs. we find actually that this relative regulatory potential is a lot stronger than the super enhancer approach. In a lot of cases, super enhancer is really just marginally above random prediction. The relative regulatory potential is a really much stronger predictor. And so this is just going back to the MARGE. But so the MARGE idea was just to calculate this regulatory potential based on signal. So what LISA is a kind of, it's a step beyond that. And so in LISA, what we want to do is to use these regulatory potentials in order to infer what the TFs that regulate the genes are. So I'm not going to go into like detail about how it all works, but the idea is that we can use this K27 acetylation data to generate a model of what the relevant data is that can be used to predict these differentially expressed genes, as well as DNA-seq data and also the transcription factor binding data. And so we have this model where we combine all of this together to make a prediction of the regulators of the genes. And then comparing with other kinds of data analysis, we see that we on a fairly large benchmarking data set, we can find that LISA is doing a lot better at predicting than other methods. And also we noticed that the Chip-seg is actually pretty good. When you have the Chip-seg, it's really a lot better than just using very kind of naive motif analysis methods. Although it might be possible that more sophisticated motif analysis methods could do better. And just one of the results coming out of LISA was that we noticed in our benchmarking data that often we had several different data sets. Some of them involved activation of a transcription factor, and some of them involved the knockdown of a transcription factor. And so what we found is that in many cases, when you have the activation of the transcription factor, LISA was finding the TF in the upregulated gene set, but it wasn't really like finding the TF so strongly in the downregulated gene set. And then vice versa, when we had the knockdown experiment, LISA would often be able to find the downregulated gene, but not the upregulated gene. And so this suggests that for a lot of these transcription factors, the direction it's one way that the transcription factor has an activating role, and that the target genes are the ones which are activated by the transcription factor, and that in a lot of cases that when you see the genes in the other direction, it might not be a direct they might not be directly related to that transcription factor. They might be some other kind of byproduct or a secondary effect. So I'm not saying that's true for all transcription factors, but I think for a lot of transcription factors, that's the case. So this is just an example. So for example, we did analysis of a MYC knockdown experiment. And so LISA finds the MYC it predicts MYC to be the key regulator for the downregulated genes. This is a knockdown, so MYC is largely activating function. And then we find a lot of other kind of transcription factors in the other direction. So the consequence of knocking down MYC might result in regulation of these other kind of modules. And then also SOX2 knockout, you'll see SOX2 OCT4 nano are highly related to the downregulated genes. Nano is also associated with upregulated genes, but these other kind of more differentiation associated genes are associated with the upregulated gene sets. So actually, okay, so when you use LISA, it's also simple to use. One tricky thing, which I hope people appreciate, is that you have the option of putting in two gene sets. So the gene set 1 is, for example, the upregulated genes, and a gene set 2 you could put in as an option as the downregulated genes. the idea is not that it's going to compare gene set 1 with gene set 2, but the idea is that you just do the analysis of these 2 gene sets at the same time, and then you have some extra features which allow you to compare the analysis. The one is not the control of the other. If you want to actually put in a custom control, then this is the place where you put in the custom control. Although I would not really, in most situations, I would not really recommend that. The control is a background gene set, which is, it's not the whole genome, but it's selected from the genome. It's a set of genes which is not really enriched in any category. It's a sort of a broadly distributed set of genes which can serve as a good background. And then, yeah, you specify like these labels, put in the job name, and run it. It will take some time, but then you just come back, you know, a few minutes later, and you can see the results. And so what Lisa shows is this ranked transcription factor. So these are ranked by the p-value for those samples. And so one interesting thing to notice is that it's ranked by different samples. So for example, in this case, SMAD3, it's this particular sample of SMAD3 which is enriched. We haven't sort of aggregated all SMAD3 samples. So you can see this is the higher ranking than that one. But for example, this beta-catenin sample here has a much lower p-value than that sample. And so then you might be interested to know, okay, you can click on this sample and you can look in the inspector and find out more about what exactly that sample is. You know, it could be that actually there's a sort of misannotation, happens not to be beta-catenin, or it could be something interesting. And you know, sometimes you see this, that if you go into the details, it's like a very particular experimental condition or something very special about that. And so that can sometimes be pretty insightful, give you a lot of insights about this. You know, it can also, it's one of those cases of the yard sale. You pick up a real gem from things from other data which you would not normally think would be useful. So if you use the option of putting in 2 gene sets at the same time, this is what you get this plot where you can look at the p-values of both. And so what's useful in this kind of plot is that you can see, for example, that this sample is really highly enriched in the upregulated genes, but not at all in the downregulated genes. Sometimes you can do these analyses, and then you see certain transcription factors which are kind of enriched in both up and down. I think it can be quite useful to look for like what is kind of more enriched on the one side than the other side, to try and understand what's going on. So Lisa, it has this analysis based on TF Chip-seg. It also has motif analyses. So the motif analyses tend to be not as predictive in general as the TF. However, I think it sometimes can be quite useful. I don't think we really understand that, but I think it can be useful to see what there is and maybe to try and think about like, why do those motifs show up in the analysis? But I would rely a lot more on the actual Chip-seg binding than the TF, than the motif-based analysis. So also we did a lot of benchmarking analysis, and so we have a lot of data. If you go to the Lisa website, you can look at the results. So we've run the results. You can look at the Lisa analysis, and you can see what kind of results you get for all of these different samples. And so that's a way of seeing what is a typical kind of results. When you do your analysis, you can do some sort of evaluation. Is this like something that you would see commonly in other data sets, or it's a good way to get some sort of intuition of what results to expect. And so also the gene set, so you can actually just download the gene sets, and it's also linked to Geo to see what exactly the experiment was. So that's actually just the kind of the summary of Lisa. And then I just wanted to just say a couple of words about what's coming to Cistrome. As Shirley mentioned, that we are doing this imputation where if you look at this plot, you can see that there's a lot of Chip-seq data out there. However, a lot of the Chip-seq data is actually concentrated in just a few cell lines. So this would be, data coming, a lot of data coming from ENCODE, for example, which is just concentrating on very few cell lines. But then, of course, there are many kind of Chip-seq data sets, different transcription factors and cell types out there. And even if you just zoom into this really like corner which looks really dense, you can see even that corner is pretty sparsely populated. And so there's a real need to try and like extend the data we have from available Chip-seq and DNA-seq into the sort of try and infer what are the other binding profiles in these other kind of cell types. And so in the Cistrome, I kind of see it as this, where you have three points of view. One is data, one is analysis, one is visualization. So currently what we've been doing is we've been doing analysis based on data and visualization based on data. But of course you can go from the other direction. You can do some analysis to sort of acquire more data. You can sort of supplement your data through analysis. The other side is the visualization has also been a sort of unidirectional thing. So we're also working on a collaboration with Niels Gellenborg lab on a visualization tool. It's interactive, where you can have visualization, but then you can sort of also do some query of the data as well as some sort of interactive analysis. And so this is still under development, and hopefully we'll soon be able to show some of that maybe at one of these meetings. So in summary, I've kind of gone over some of the main functionality of a Cistrome data browser. Firstly, there's the Cistrome data browser, which is this collection of chromatin profiles in human and mouse. You can search in various ways by keywords. You can quickly evaluate the quality of the samples, and there is some downstream analysis of the motif and the target genes. I've also described the toolkit. It can answer what regulates a gene of interest, what transcription factors bind in a genomic interval of interest, and also what cistromes overlap with your input cistrome of interest. And then there's a Cistrome Go, which is a sort of amalgam of beta and great. Although it doesn't use the same methodology as great, what are the genes directly regulated by a transcription factor? What is the function of the genes regulated by a transcription factor? And then finally, Lisa, which answers the question of what transcription factors regulate a differentially expressed gene set. And so yeah, thank you, and thank you. I thank all the people who have worked on this project, Shirley and Miles, and Henry, and Shirley's group, a lot of students in Tongji, and also recent collaborators, Niels Gellenborg's group. And I'll be happy to take any questions.
Thank you, Cliff, for that summary. I want to make a comment about Lisa input. Basically, we look at the differential gene expression and you input that gene list into Lisa. Usually, we want this list to be not too small. For example, if you only have 5 genes that are differentially expressed, it's very difficult for us to figure out the lineage. But if you have like a few hundred genes that are differentially expressed, we almost, based on the differential gene list, we can almost guess what cell type it is, what lineage it is from. And that's kind of what helps Lisa to build the epigenetics model. But at the same time, we don't want this input gene list to be too big. You can imagine if you have like 2,000 differentially expressed genes, when you input Lisa, it will only give you more lineage specific things, which may or may not have anything to do with the specific differential expression treatment conditions. And so the correct number of genes to input into Lisa is kind of important. And we usually recommend between 200 to 500 genes to run Lisa.
Right. Yeah, right. So yeah, so I think, if you have 50 genes, it's kind of probably, I definitely wouldn't try less than 50. Definitely 100 or 200 is much better.
Yeah, so Cliff, I wonder whether we should update the main website when people enter the list, give them that cue to show them that the input list should be, you know, yeah, I think ideally between 200 to 500.
Right.
Thank you for this. I was wondering if you could say something about the export of the data results.
So, for example, I think most of the tools for example, Lisa, you can download all the results, and I would recommend that because we leave it up on the website, but probably for a month. We're not going to keep it forever, but you can download the files. And I think for all of these analyses, you can download the results.
Okay.
Yeah.
Thanks.
Thanks, Cliff.
Okay.